they're going to be your best phagocytic cells in your body, and they're going to be the ones who actually start inflammation. Because remember, when you begin studying Chapter 3, macrophages can recognize stuff through their PRRs, right? Who can tell me what a PRR stands for? Pattern recognition receptor. Awesome. Pattern recognition receptor, right? And PRRs recognize what? PAMPs, what does it stand for? You remember? Or anyone else? Pathogen? Associated? Something pattern, molecular pattern, right? So you can recognize, for example, LPS with TLR4. And then you had like a whole intracellular cascade you had to study with MID88 and other proteins. And then NF-kappa B, which is a transcription factor, would go into the nucleus, activate your cytokine genes, and then your macrophage would start secreting TNF gamma, IL-1, IL-6, IL-8, IL-12, and then all those would cause increased vascular permeability, vasodilation, pain, and influx of leukocytes, right? And that's how you would fight whatever infection was coming to, attack you. Any questions so far? Oh. Actually do this and then. All right. So which of the following lymphocytes has orange-red stained granules and is specialized in fighting parasitic infections? Is it a basophil, a neutrophil, a B cell, a eosinophil, or none of the above? How many of you say eosinophil? How many of you say another answer? Why is it another answer, the correct answer? Yeah, what's up? It's asking about lymphocytes, yes. How many times have you seen this question in the tests? Like he asked a question like this at least twice, right? Yeah? So as you're taking this test, I want you to make sure that you highlight keywords, okay? So when he's asking which of the following lymphocytes, go ahead and highlight it or underline it or whatever it is. And then you think about, what do I know? Which cells I know are lymphocytes? There's 3 of them. BT and NK, right? Now, are any of those involved in fighting parasitic infection, and do any of those have orange, red stained granules? No, right? Those would be eosinophils, technically, but these are not lymphocytes, so then the correct answer is none of the above. All right? Make sure you know your cells when, as you're, before the test, just because it's useful, right? Because you can have a question like this. And knowing the cells is really easy, right? You have BT and NK, those are your lymphocytes. And all lymphocytes are leukocytes, right? But not all leukocytes are lymphocytes. Because lymphocytes are a subcategory of leukocytes. Leukocytes just refers to white blood cells. And then the rest of the cells of your innate immune system are just going to be your Ben, mast cells, macrophages, and monocytes. And that's it. And dendritic cell. That was the last one. So Ben, mast cells, dendritic cells, monocytes, and I forgot one. Let me write it down. So you have B, T, and NK, which are your lymphocytes, and then your leukocytes, right? Lymphocytes, and then your leukocytes are Ben, your macrophages, your dendritic cells, and then your mast cells. Cool. Make sense? You can pull this up from the hematopoietic chart from like chapter 1 or 2. Okay? Cool. Any questions so far? No? All right. So then pyogenic refers to the body's production of heat to fight infection. Is this true or false? False, right? Because pyo means pus. Pyro with an R is the heat production. The way I remembered this is for pyro, I would think of a pyromaniac, someone who likes to light things on fire, right? So that's the heat production part of. And then the pyo is, I would always say, yo, that's pus, and that's how I would remember it. But you can do whatever mnemonic works for you. No. Cool. Now, complement. The protein that initiates the cascade in the classical pathway is going to be C1q, C3b, C1s, A and C, or none of the above. Which one's the correct one? What's that? She's saying C1q. What about everybody else? What do y'all think? C1q or C1? I mean, we're going to be split between C1q and C1s because we know C3b doesn't, I mean, it's involved in the alternative, not the classical. But what about C1s? How many of you think C1s is the one that activates complement? Here's one. Okay. So the correct one is C1s, actually. And I'll tell you why. So let me just... I'm going to upload this online, by the way. So I'll write down the questions. This question 2. I'll do that, and then so that you know the sketch it refers to which question. 4. Oh, I broke it. I fixed it. Okay, so alternative pathway. Remember, it starts on the surface of a pathogen. So let's say this is a Staph aureus, just to give it a name, right? This is the surface of the bacteria right here. IgM comes in, right? IgM is a pentamer, so there you have your 5 little arms. And then C1q comes down onto IgM. And remember, it doesn't have to be IgM. It can be 2 IgGs or 1 CRP, right? But you always need something to bind on the pathogen first, and then C1 comes down on it and binds to IgM, IgG, CRP. So then you have C1q coming down like this. And then here C1q had, C1q had 2 subunits, if you remember. It had C1R and C1s. Let me just change the color here. So when C1q binds to, all right, so C1q is green, C1R is blue, and C1s is brown, right? So C1q, and the 3 of them make the protein C1. So basically when C1q binds to IgM, that C1q activates C1R, then C1R activates C1s, and C1s is the part of the protein that has what we call the catalytic function. So it's the one that actually does all the cutting. So because of C1s, you're going to have C4b cut into C4b A and B. Sorry, C4 into A and B, and then the same thing for C2, right? So then thanks to this, you're going to have C4, and then the same thing for C2. C4b. Make sense? And then what makes the convertase, the C3 convertase of the classical pathway? C4b? 2a, right? So then you're going to have C4b and then C2a coming down, and they form C4b2a, which is the classical convertase, right? And then this is going to cut C3 into C3a and b. Yes? And then what happened? C3b can do how many things? Yeah. Sure, go ahead. Tell me which ones. One. Do you know one at least? Or someone else? It doesn't matter. It doesn't have to be you. Thank you, though. Anyone? Oh, yeah. It opsonizes. Awesome. So then receptors can recognize C3b, such as which receptor? CR1. Cool. And what's another thing that C3b is really good at? Or that it can turn into, I guess? It can turn into a C5 convertase later on, but before it turns into a C5 convertase, it needs to turn into a C3 convertase, right? So it can turn into a C3 convertase and then into a C5 convertase and, poke the hole in the bacteria. Does it make sense? Cool. And then C3b, let's just draw it really quick, right? C3b can come in along with factor B, so that, and then you have factor H and all that. I'm just skipping steps so that we can get it through the whole thing. But you basically, in the end, you end up getting C3b, big B, little b, which this is your alternative C3 convertase, right? And this can also cut C3 into C3, A and B, and so on. But then this can also turn into a C5 convertase as well, right? Ooh, what did I do? Convertase. Cool. Any questions about complement? No? Make sure you know complement. Like, it's the easiest, in my opinion, it's the easiest thing that you can know so that you can get more points on this final exam, right? Because complement has been coming back on every test, I think, or at least 3 out of the 5. So if I were to bet money on which questions are there going to be or which topics, this is one of the ones that I would bet most money on. And it's really not that bad, right? You just have to know, you have to memorize the proteins, C1 through C4 technically, and then, because you don't really have to study the MAC, all you have to say is that C5 through C9 forms the MAC, and that's it. And boom, you know complement, right? Any questions? No? Okay, cool. Let's draw a line here. So next question is, the receptor for C3b. We just said this. Which one was it? CR1. Awesome. Now, this next question is about the Tolllike receptor. So this is going back to chapter, I want to say 3, where you had the macrophages with its PRR recognizing stuff, bacteria or whatever it is, so that an immune response can happen, so that the macrophage starts the inflammation through the release of cytokines. And how do How do you signal the macrophage to start making and releasing cytokines? Activating a Tolllike receptor, right? So this question is just asking about what extracellular proteins Tolllike receptor need to be able to start the cytokine producing protein cascade. And the correct answer is that it's all of the above. This is a little too specific. This is one of those things that I don't know if you would ask a question like this, but I just told you, so if you can remember this, then you're good to go, right? But just in general, remember the concept that macrophages use their PRRs to recognize PAMPs so that they can get activated against an infection, and as a result, they'll be secreting cytokines, right? TNF alpha, IL-1, IL-6, IL-8, and IL-12. Okay? Cool. And what, oh no, this is not good. Delete, there you go. Yeah. And then what happens as a result of the secretion of those cytokines? One word. Inflammation, yeah. And then you have the influx of leukocytes, vasodilation, and all that good stuff. All right? Cool. So this next question is asking to identify the mismatch pair. And this is just going into protein binding, right? So adhesion molecules and other signaling proteins that bind to each other. So let's go really quick. So ICAM1 and LFA1, if you go and check your adhesion molecule charts, these just don't bind to each other. Okay? CD14 and LPS, I'm sorry, they do bind to each other, my bad. One is a immunoglobulin-like superfamily, and then the other one is a, I forgot the name of it. But both are involved in the tight binding of a neutrophil, if you want to go back and review it. Then CD14 and LPS, they do bind to each other in the Tolllike receptor area outside, so that if you want to review that, it's on the macrophage, it's chapter 2. NK cells and interferon gamma, yes, because those are the ones, NK cells secrete interferon gamma and receive interferon gamma to be activated. And then selectin type binding, this is the one that's wrong. It's the one that's mismatched, right? Because selectins are involved in rolling adhesion, right? So remember, selectins don't have, they will not give you tight binding. They're not going to be that strong. They're not going to be as strong, for example, as ICAM1 and LFA1, right? And selectins bind to vascular addressins, just so you know, okay? Any questions? If you have any questions, don't be afraid to stop me or raise your hand, please. We have, hour and a half still. Cool. So catalase is the enzyme produced by neutrophil to produce hydrogen peroxide. Is that true or false? False, right? It actually, catalase breaks down hydrogen peroxide. Why is it good that we have cells that break down hydrogen peroxide? Does anyone know? Yeah, it's really bad for us. It can produce oxygen radicals, and those are not good for you. They'll destroy whatever cell they're nearby. That's why if you ever, don't do this because it burns, but if you ever have a cut in your hand or something and all you have is hydrogen peroxide and you squirt it on it, you're going to have little foam forming. That's the catalase from the neutrophils working, basically, breaking down the hydrogen peroxide. All right, so this 8 is false. Everybody back there can see well? Yeah? Okay, cool. If anything, you could even, if you can't see well, you can even open up the stream on YouTube and look at me through there in your computer if this doesn't work. It's going to be a little delayed, but you're going to be able to see at least. Sorry for the weird setup. I apologize. It's weird. Anyways, interferons. interferon alpha and beta are produced by which cells? What did interferon do for our cells? Let's say an epithelial cell. When would an epithelial cell in like my sinuses would produce interferon alpha and beta? Does anybody remember? So interferon alpha and beta was made whenever you have a viral infection, right? You start making interferon beta first, which is secreted. It's like an autocrine secretion, so it's secreted onto itself, so that interferon alpha is made. And basically what they do is that they'll induce changes in the cell so that you can slow down viral replication. And you also help out with, you facilitate MHC1 presentation so that your T cytotoxic cells can come in and kill you, basically, as an infected cell. But the question is, which cells make interferon alpha and beta? You had two classes. Class 1 is interferon alpha beta. Class 2 is interferon gamma. Does anybody remember this? No? Okay, it's fine. I'll tell you. Interferon alpha and beta, they're made by all nucleated cells. Okay? Whereas interferon gamma, which is that class 2, is made by NK cells. All right? Any questions? No? Okay, that's fine. Also, with interferon alpha and beta, your cell, because remember, you only make this when you are a cell that's infected by a virus. So you'll also put out proteins, basically signaling proteins that will help to signal the NK cell, hey, I'm infected, come kill me, basically, right? All right. So antibodies, binding sites. So let's draw this really quick. So this is question 10. Let's just do it here. All right, so antibodies. Let's say this is a BCR, right? So the green here is going to be your heavy chain, and your red is going to be your light chain. Okay? So let's do... It's not the same type of green, but it'll work. So this is light chain and heavy chain. So really quick, for the heavy chain, let's talk about the DNA first. What are the genes that code for your heavy chain? V, D, and J. Remember that, okay? So you have your V, D, and J. And then for your light chain, what are your genes? V and J. Cool. Now let's talk about isotype. What are the isotypes for the heavy chain? There's 5 of them. Yeah, gamed. G, IgG, IgA, IgM, IgE, and IgD, right? Which one's the first one that we do all the time? IgM. And then we can isotype switch to whatever it is. If you're a B2 cells, what about B1 cells? Are B1 cells able to isotype switch? No, right? So IgM is the only thing that B1 cells will make. So heavy chain is G, A, M, E, and D. And then M is the first one. Produced, right? Now, what about the light chain? What are the isotypes of the light chain? We didn't talk about it too much, but there are two isotypes for light chains. Yeah. Yeah, kappa and lambda. Remember? So you have two isotypes for your light chain as well. But doesn't matter the isotype for both heavy and light chain, the genes for them are always going to be VD and J for heavy chain, V and J for light chain. So kappa and lambda. All right? So really quick, now talking about, well, I guess I'll leave it like this so that you can still see it. One monomeric antibody, how many binding sites will it have? So this thing right here, how many binding sites does it have? Not a trick question. 2, right? There's 2 binding sites to it. Each binding site is made by one heavy chain and one light chain. Yeah? Now, each heavy chain and each light chain, right? Basically the top here, these top boxes, if you want to call them that way. This is where the VD and J and V and J comes into play, right? But what I want you to take out of this is that each heavy chain and each light chain on each side, they will have 3 hypervariable regions each, for a total of 6 hypervariable regions, or CDRs. HV and CDRs are the same thing. So I'm going to go ahead and write it here. So the heavy chain is going to have 3 HV, and then also 3 hypervariable regions, for a total of 6 hypervariable regions. All right. Does it make sense? Yes? So how many hypervariable regions or how many CDRs will you have in one binding site? 6, right? How about in one antibody? That's so like secreted IgG, which is one. This could be secreted IgG, right? How many will you have total? 12, right? Do you all agree? What about, so then let's go into the dimeric and pentameric. So what about a secreted IgA? How many hypervariable regions will it have? Because IgA is a dimer, so it's going to have 2 of these, right? So 12 times 2, 24, right? And what about IgM? Secreted IgM has 5 of these. 12 times 5, 60. Cool. Awesome. So just remember, like if you were to come across a question that talks about how many hypervariable regions do you have in a antibody, blah, 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 blah. Just remember, 6 hypervariable regions per binding site, and the minimum binding sites that you have on an antibody is 2. Then you have to figure out whether it's monomeric, dimeric, or pentameric, right? Monomeric is IgG. It could be IgA, because IgA has monomeric and dimeric. And then IgM has 5 IgMs together, which is a total of 10 binding sites, which is a total of 60 HVs, hypervariable regions. Make sense? Yeah? Cool. All right. So then, one binding site of any immunoglobulin is composed by 6 hypervariable regions. Yay! All right, so This question again, I sprinkled the exam with a couple of specific questions that, in my opinion, these are like good things to know that are specific, but they're still, they still apply for the test. That if you know him, good, right? But if you don't know him, I don't know if he's going to ask him or not, because they're, like I said, they're a little too specific. But this is just talking about hypervariable regions, right? So in one heavy chain, for example, you have the hypervariable region 1, 2, and 3. And 1 and 2 for both heavy and light chain, they're always going to be coded by your V segments, okay? And then your third hypervariable region in the light chain is going to be coded by the junction of V and J. And then in the heavy chain is just the junction of V and D, and then the junction of D and J. All right, so just remember, CDR is 1 and 2, always by the V segment in both heavy and light. And then CDR3 for light chain is just the junction between V and J. And then in heavy chain is going to be a junction of V and D, the D segment, and then the junction of V and D and J. Okay? Cool. So then the correct answer here is going to be C, the V and J junction. What's up? Yeah, I'll write it down. So, so for your heavy, so let's see, CDR. CDR diversity. So for the heavy chain, right? For the heavy chain, your CDR1 and CDR2, these are going to be coded by your V segments, right? And then your CDR3 is going to be coded by the VD junction plus the D segment. And when I talk about the D segment, it's just this, right? When we talk about V, D, and J, remember, like, you have a V that's, so going back to somatic recombination really quick, you have a bunch of, for the heavy chain, you have, 20 Vs, 20 Ds, and 20 Js. And to make an antibody, you only need one of each selected at random by RAG1 and 2. So RAG1 and 2, they do the cutting of each segment, and then TDT is the one that adds random nucleotides in between so that the DNA can be literally glued together again so that they make one DNA strand, and you can transcribe it, translate it, and make a protein. So when I talk about the junction, it's just those random nucleotides that are being added by TDT in between V and D, or D and J, or V and J. And then when I talk about the segment, is just this, or this, right? D or V itself. So then going back here, the D segment plus your DJ junction, right? And then for your light chain, so your CDR1 is going to be, 1 is also going to be your V segment, and then CDR, 1 and 2. And then CDR3 is going to be your VJ junction. So then the most diverse part of the binding site of an antibody is going to be the CDR3s for heavy and light chain, because they're made by the junctions of the gene segments, and those junctions are just going to be at random, because that's where TDT just adds random nucleotides in between. And that's the part that if you were to compare, the DNA that you have for an antibody compared to the one of your parents, that's where it's going to be completely different, because that's the whole recombination of DNA where you add random stuff in between. Make sense? Cool. Any questions? No? All right. So then, whether an antibody is secreted or membrane bound, this is determined while or during somatic recombination, somatic hypermutation, RNA splicing, DNA splicing, or ANC. So this is, again, part of the whole somatic recombination. And the correct answer here is RNA splicing. And yeah. So I'll go ahead and change this on the answer key, because technically you can choose membrane bound and secreted at any time. So it can be during somatic recombination, and it can be during somatic hypermutation, just so you know. But the important thing is that you decide whether you will put something on your membrane or you're going to secrete it during RNA splicing. Okay? So the B cell will always, as it's making more antibodies, it's going to be able to decide, not during the DNA editing part, but during the RNA editing, it's going to be able to decide whether, all right, am I going to secrete this, right, or am I going to put it on my surface? When it's going to secrete the antibody, it's probably going to be a plasma cell. And when it's going to put it on its surface, it's probably going to be either a maturing B cell, a cell that's going through somatic hypermutation and it needs to put those new improved antibodies on the surface, or even a memory cell that was just a plasma cell that got deactivated and told to go back into the memory cell stage. Does it make sense? Yeah? Any questions? No? So I'll make sure that on the answer key it just says A, B, and C, and I'll put like a little note to the side that explaining why. What's up? This is going to be the somatic recombination chapter. I want to say it's like 4. Hold on, let me see really quick. Lecture. Yeah, chapter 4. Let me just make sure. Yep, antibody structure and generation of B cell diversity. So I can actually pull up the slide really quick. Here it is. So I don't know. Oh, okay, cool. So you can see here that, oh, wait. Anyways, here in the, I guess I have to do it this way. I don't know how this works. I'm just going to point at it this way. So the heavy chain is the one that binds to the surface of your B cell, right? So during the mRNA sequence, where you do the splicing, you can actually select here whether to leave the bottom part that attaches to the cell membrane of the B cell, or take it away and make it a secreted mRNA. So basically the takeaway is just to know that deciding whether it's secreted or membrane bound, mRNA splicing or RNA editing. Okay? Yeah? Cool. Cool. All right. So Next question is still, talking about your the B cell and BCR, but this time it's talking about hypermutation instead of recombination. So it's asking basically the enzymes involved in somatic hypermutation is or are RAG1 and 2, AID, RNA polymerase, C and D. Which one is it going to be? AID, right? AID is the one enzyme that will turn C's to U's, right? And then another enzyme is going to come in and it's going to realize, oh my God, there's uracil in the DNA. What is going on? And they're going to fix that mistake really quick. And they're going to put in a random nucleotide in there. It can be, again, a C. So then, you try to do a mutation, but it didn't work. Or if you change it to something else, then you're going to end up with a binding site that's either the same, better, or worse, right? And then in the end, because of the way somatic hypermutation works in the follicle, you're only going to get a B cell that made better antibodies that bind more strongly to the antigen. Make sense? Any questions? No? All right. So T cells. So once the T cell has rearranged the TCR, which of the following membrane proteins will the TCR need to leave the endoplasmic reticulum to get to its final destination, which is the T cell surface? What are the co-receptors that the T cell will always need? What's that? CD3 complex, right? And the CD3 complex consists of 2 epsilons, 2 zetas, gamma, and delta, right? So 2 epsilon, 1 gamma, 1 delta, and 2 zetas. Yeah? Cool. And remember, there's a question. I was going to say it, but let me ask it. So the importance of the zeta chain in the TCR is that they play a role in stabilizing the TCR. Then B says these proteins have a role in transducing signal from the extracellular space to the intracellular space. C says that there's no role in aiding the TCR when interacting with the MHC. And then D says without zeta chain, ZEP70 would not phosphorylate and activation of T cell would be impaired. So then the correct answer is going to be E, right? Because they have a really important role in transducing, so basically sending the signal from the extracellular space, meaning the signal that the TCR receives when it recognizes something on an MHC, to the intracellular space, which is where you have all the proteins, the whole protein cascade that you've studied of ZAP70, phosphorylating PLC gamma, and it turns into DAG and IP3 and so on and so forth. Do you remember that? Okay. So that is not going to happen if you don't have zeta chains, basically. All right, and just to refresh your mind, this is what I was talking about. I can also, this is on my ucfsi.wordpress.com. You can pull it up from there. But this is it. So this is these are your CD3 complexes. So you're going to have 2 epsilon, 1 gamma, 1 delta, and then these are the zeta chains. And then the green little dots are phosphorylations. And then you have LCK, ZAP70, and then PLC gamma is activated by ZAP70, and then PLC gamma breaks up PIP2 into DAG and IP3, and then DAG and IP3 will activate transcription factors, and then you have T cell proliferation, and so on and so forth. IL-2 secretion, and so on. So that's like half of chapter 7, I think. Right? If you know this for the test, great. Like I said, it's a little too specific, so I don't know if memorizing each step is going to be beneficial, but at least understand the concept of how T cell activation works. What are the key proteins evolved, right? So at least knowing this outside would probably benefit you. All right? Any questions? How many of you are taking the test tomorrow? Okay, a couple of you. Cool. So make sure you get a good night of sleep today. If you're taking it Tuesday, make sure you get a good night of sleep on Monday, right? But yeah, you guys will be fine. You'll see it's really not that bad. So MHC1. MHC1 is, before we go over this, what kind of peptides will you load on MHC1? Intracellular or extracellular? Intracellular, right? So how many, or which type of cells in your body will be able to have and make MHC1? Anyone? What's that? All nucleated, yeah. CD4 fall under that category technically, right? So all nucleated cells will be able to make MHC1. Yes? Y'all remember that? And that's because all nucleated cells can be infected by viruses, right? So when you're infected by a virus, you have those mechanisms that start working, and some viral proteins will be loaded on an MHC1 molecule, put it on the surface, and then CD8 cytotoxic T cells will come in, recognize whatever you presented, and then secrete cytokines so that you're told to die by apoptosis. So that you avoid, as a cell, you avoid bursting into a bunch of little virions and infecting the cells around you, basically. Make sense? Yeah? This question mostly is talking about the structure of MHC1. So it says MHC1 has 2 transmembrane chains with 2 alpha chains and 2 beta chains, or 1 transmembrane chain, 3 alpha chains, 1 beta 2 microglobulin, 1 alpha 1, 1 alpha 2 that will form the peptide binding site, and then a beta 2 microglobulin that also has a transmembrane chain. So let's draw it really quick, and I can draw also hold on, let me see. This is question 16. We can do it here. So MHC1 is going to look like this. Alpha 1, alpha 2, alpha 3. The only transmembrane chain, which refers to which part of the protein binds to the cell, is on this 3rd subunit of MHC. Okay? And then the peptide binding site is formed by alpha 1 and alpha 2. All right? I think it's really helpful to know for exam. It's really helpful to know structures. You don't have many structures that you need to know, so it's BCR, TCR, MHC 1 and 2, and that's it. That I think would be helpful, like on a test, that Professor Wiggle could ask questions about it. So just know how to draw them, know like each subunit, how you call them, and then knowing like which specifically subunit, which one does it have the peptide binding part, and which one is the part that binds to the cell. I think those are helpful things to know, all right? And then this is the beta 2 microglobulin. And then MHC2 looks like this. You have alpha 1, alpha 2, beta 1, and beta 2. All right? So then the correct answer is going to be E. So basically, MHC1 has 1 transmembrane chain, 3 alpha chains, and 1 beta 2 microglobulin. And then also oh no, never mind. Woo! So this has beta-2 microglobulin that also has a transmembrane chain. That is wrong. The correct answer is B. My bad. Make sense? Cool. All right, so question 17 says, the maximum number of HLA genes that Professor Weigel could receive from his parents in the event that he is homozygous for MHC1 and heterozygous for MHC2 is going to be how much? So if you're heterozygous for both MHC1 and 2, how many can you have total? 12, right? 6 MHC1s, 6 MHC2s. Now, if you're homozygous for let's say MHC1 in this case, right? How many MHC1s will you have if you're homozygous for MHC1? 3, right? Because if you're heterozygous, you could have 6. Homozygous is half 3. And then, in this case, Professor Weigel is still heterozygous for MHC2, so he gets the 6 for MHC2 for a total of 9. Does that make sense? Everyone? Yeah? Okay. So basically, the minimum amount of MHCs that you could have if you're homozygous for both, you're going to have 3 from MHC1 and 3 for 2, for a total of 6. Then you could be homozygous for one and heterozygous for the other one, so 3 and 6, 9. Or heterozygous for both, so 6 MHC1s and 6 MHC2s. Question? No, it's not B and C because, oh yeah, my bad. It is B and C. So when I read, for some reason I read B and D it's wrong, but it is, yes. Good call, thank you, appreciated. Here it is. But going back to the amount, like the number of genes of isotypes, if you will, or allotypes that you can have. Does it make sense? How many can you have total, depending whether you're homozygous or heterozygous? Yeah. Yeah, that's fine. So if you're homozygous, meaning that you got the same allotypes from mom and dad, for MHC1 you're going to have 3, and MHC2 you're going to have 3. So if you're homozygous for both, you get a total of 6, 3 and 3. Then if you're heterozygous for both, you can have 6 from MHC1, 6 from MHC2 for a total of 12. And then you can be like homozygous for one and heterozygous for the other one, so it's 3 and 6, 9. Does it make sense, Al? Yeah. Yeah, just always think the minimum, when you talk about both MHC1 and 2, the minimum you can have is 6 if you're homozygous for both, right? Because you have 3 for MHC1, 3 for MHC2. And then for, if you're heterozygous for both, you're going to have 6 and 6 for a total of 12. And then you can just be, it can happen that you're homozygous for MHC1, for example, and heterozygous for 2, or vice versa. So then you're going to have 12. No, you're going to have 9 instead of 12 or 6. All right? Cool. So a mutation in the MHC locus will affect the beta-2 microglobulin gene, which is not true because this question taps into MHC1 loading and production, where you had to study like the whole TAP1 and 2, calnexin, calreticula, and ERP1, and so on. That's chapter 7 of the T cells and of TCR and MHC. And this is just to remind you that the beta-2 microglobulin, so this right here, this is added after you make your MHC, after you, as a protein, you make, as a cell, you make alpha 1, 2, and 3. And it's just added for stability, right? So if you were to mutate MHC1, the MHC1 genes, you're not going to affect the beta-2 microglobulin because it comes from a whole other protein, basically. The beta-2 microglobulin is just something else, a different protein that it's added to MHC for added stability. And it's added after you go into the whole DNA and all that. So it's not even, they're not even close. Does it make sense? Cool. That's another, oh Lord. That's just another question that's a little specific, but I think they're useful things to know that they might pop up. Hopefully they pop up and you already know it, right? So question 19 talks about the TCR and the MHC interacting. So I'll read it to you and then we can go over it really quick. So when interacting with a TCR, the peptide loaded on the MHC will mainly interact with the blank region of the TCR, while the blank and blank regions will interact and recognize the MHC. So remember, the TCR, no, this is question 19. So this is your TCR, right? Here you have your CD3 complex, and then your zeta chain, right? This is alpha and beta. And then the MHC comes up from, like, let's say down here with a peptide. Cool. So if we were to zoom in into the TCR, remember the TCR also has VDNJ and VNJ genes, meaning that they will have CDR regions, just like the beta chain, right? CDR is 1, 2, and 3 for each, right? So you're going to have 3 on the alpha chain, 3 on the alpha chain here, and 3 on the beta chain here. Just remember that CDR, I'm going to zoom, CDR1 and CDR2, they will recognize the MHC, the sides of the MHC, and then CDR3 will recognize the peptide, basically. Okay? Why is that? CDR3 is the most variable one, so there's more chance for it to recognize something very specific, a peptide. Versus CDRs 1 and 2, they're not so variable, and they need to recognize something that's always the same, which is an MHC, because MHCs, we only have a few molecules in our body. We can't make that many, right? So this is just to remind you that CDRs 1 and 2 recognize the MHC, CDR3 will recognize the peptide. And that's true for alpha and beta chains. Okay? All right, so one, two, and 3. So the correct answer is B. All right, so who can tell me a little bit about SID? Does anybody remember SID? That's one of the diseases that we have to study. Anyone? Severe combined immunodeficiency. And what was affected in SID? Anyone? The RAG1 and 2 genes? Yeah? So what happens if you don't have RAG1 and 2? Yeah, you don't have somatic recombination. If you don't have somatic recombination, what are you not going to have? What's that? you're not going to have T cells or B cells. So then the correct answer here is B. So TCR recombination is affected. T cells are not functional because, because with SID, TCR recombination is affected. Make sense? Cool. All right. So little Jimmy was taken to the doctor because of an upper respiratory infection that he does not seem to be able to clear. A differential blood count showed that Jimmy had a normal lymphocyte count, but then when they did a genetic mapping of his HLA genes, so basically the MHC genes, he showed several mutations that affected his TAP proteins. Most likely he will have what? So if you don't have TAP proteins, you're not going to be able to load MHC 1 and 2, because remember, TAP proteins they work sort of as a gate between the cytoplasm and your ER, so that peptides can be transported from the cytoplasm to the ER and loaded on an MHC class 1 for presentation. So I just told you the first part of the answer, right? And then the second part is that he has bare lymphocyte syndrome. Make sense? Yeah? All right. Cool. So HLA-A, is that a type of isotype? What is it? Is it MHC1 or MHC2? MHC1, and you also have other types of HLA-As, right? You have HLA-B, HLA-C, and so on and so forth, depending on how many, if you're heterozygous or homozygous for it. So MHC1 presents to B cells, or CD4 T cells or CD8 T cells? CD8 T cells. So then the correct answer here is going to be B, because it presents to CD8 T cells, and then that it's a type of MHC1. Other types include HLA-B and HLA-C. I just realized I have a typo here. I'll make sure I fix it later. Cool. Any questions? Then remember, for MHC type 2, you had HLA-DR, DQ, and DS. Those are the human types of MHC, of MHC2. All right? So now B-cell maturation. Who can tell me the stages of B-cell maturation really quick? Anyone? No one? Was it pro or pre first? Pro, right? So you have early pro, late pro, large pre, small pre, immature, mature. Right? Cool. Which stages do you work on your heavy chain? In the pro or the pre? Does anybody remember that? Which stages does the B cell work on the heavy chain? As a pro or a pre-B cell? Pro, right? Because you begin with a heavy chain. Then, so in the early pro, basically you're doing the D and J recombination. Then late pro is V to DJ. Then in the large pre, you're checking if the heavy chain you just did works, right? With the pre-BCR, the surrogate light chain. And then small pre, you work on the light chain, the V and J recombination. Then as an immature cell, you start making those BCRs that you made. You start putting out IgM first. And then as a mature cell, you're going to need to have both IgM and IgD on the surface, right? So if you don't have IgM and IgD on the surface, you're not going to be a mature B cell. Cool? All right. There you go, a large pre-B cell. So large pre, this is the stage that I told you that is testing the heavy chain. A large pre-B cell is distinguished by a pre-B cell receptor which consists of a completely functional heavy and light chain with its Ig alpha and beta on the side. This is not true because the large pre-B cell only has a functional heavy. The light chain, we haven't touched it yet. Right? So A is wrong. Then a pre-B cell receptor consisting of a heavy chain and a surrogate light chain, which is with its Ig alpha and beta. And then the surrogate light chain that tests whether the heavy chain will be able to bind to the light chain. So B and C are correct, basically. So the function of the surrogate light chain is to work as a fake light chain, right? That will bind to the heavy chain and just make sure that the heavy chain can work as far as the structural part of the heavy chain, because the heavy chain actually holds the light chain, right? So if that works, then you're good to go, and the B cell can start working on the light chain. And always remember that you have to have these Ig alpha and beta on the side all the time for that BCR to be on the surface, right? Because the Ig alpha and Ig beta, they work like the zeta chain for a T cell, and they're the ones who will do the signal transduction from the extracellular space to the intracellular space. Any questions? No? Okay. I'm going to try to power through it a little. Now, if you have any questions, stop me, because I can always upload the answer key to this, and I'd rather answer questions you have now versus, not answering them and just going through the whole thing. If we don't have enough time and I can't finish it, like I said, I have the answer key. I'll have it online as soon as I get home, so don't worry about it. You'll be able to get that, okay? So, oh, this is another question. We already answered this from, I don't know, one of the first few questions, but basically it's asking, a mature B cell will choose whether to produce IgM or IgD through mRNA splicing. So this is the other thing that you can choose as a B cell to do through the mRNA. One of the things was secreted or membrane bound. The other thing is IgM and IgD. Those are the two isotypes that your B cell will be able to switch back and forth for production without having to go into isotype switching and somatic hypermutation. Because then once it switched to, let's say, IgG, for example, the M and D genes are lost, and you're not able to make any M or D anymore. So there's no switching of M and D and the mRNA. All right? Yes? No questions? No? So the pre-BCR is also known as the, how many checkpoints do you have before the pre-BCR? Basically where you have the surrogate light chain. Do you have any other checkpoints before that? No, right? So it's going to be the first checkpoint, which is a positive signal that prevents apoptosis. So basically, the maturing B cell, when it's going through somatic recombination and it's testing its heavy chain, right? Its default is to go into apoptosis, unless it's able to test the pre-BCR, right? The heavy chain with the surrogate light chain, and it works, and thanks to that, you're going to have a positive signal that prevents apoptosis, basically. First positive prevents. There it is. Make sense? Yeah? Okay. So a translocation of the MYC genes will give you superpowers? No. Unfortunately, you get cancer whenever you have mutations. Comics lied to us. You don't really get superpowers. But anyways, you get Burkitt's lymphoma. Yes? This is a very straightforward question. This is one of these type of questions that you either know it or you don't. My suggestion to you is there's not many diseases that you need to study because this is not like physio. So just make a list of I think it's less than 10 diseases you need to know. and just write down the causes and what happens as a result of in like the immunology aspect of things, right? This is one of those. Burkitt's lymphoma, MYC translocation. Yeah? Cool. B1 cells. So the B cells that you've studied throughout the semester that go through somatic recombination, somatic hypermutation, isotype switching, and all that, those are all B2 cells, right? We also have B1 cells that are a little different. B1 cells are prenatal, so they're the type of B cells that you have the most when you're a baby in mom's womb. And they're special because they're not able to make BCRs that are, their BCRs are not going to be as diverse as B2 cells, right? What gives the diversity to the BCR in B2 cells? What gives the junctional diversity? Which enzyme? What is it? What is it? TDT. Thank you. So it's TDT, right? Remember, like I was saying, I showed you the CDR diversity, right? The binding site. The junction, the VD junction and DJ junction, and then V and J junctions. That's just referring to those peptides added at random in between each gene segments that were cut by RAG1 and 2, right? So because your B1 cells, they lack TDT, or it's expressed at a lower rate, they're just not going to be as diverse. So the main difference is that TDT is not expressed in the prenatal period, so you're not going to get, in B1 cells at least, so you're not going to get that much diversity. So they're good, but they're not as good as B2 cells, basically. Cool. So plasma cells, they can differentiate directly from, can it differentiate directly from an activated B cell? So can an activated B cell turn into a plasma cell right away? Yeah, right? Cool. Can an isotype switched B cell turn into a plasma cell? Yes. Can a hypermutated centrocyte, so basically a centrocyte that went through hypermutation, somatic hypermutation, can that turn into a plasma cell if told to do so? Yeah. Can a memory B cell turn into a plasma cell right away? Yeah. So all of the above. Make sense? What do plasma cells do? They secrete antibodies, right? So basically it's like a buffed up B cell. It's like really big. There's a bunch of cytoplasm and ER in those B cells, and they're going to be making a lot of protein. There's going to be a lot of antibodies getting secreted all the time by these plasma cells. Make sense? Cool. And then the plasma cell can just, be turned off into a memory cell, and then a memory cell can be activated again, and it can, turn into a plasma cell again, or be told, hey, go into somatic hypermutation, make an even better antibody, right? Cool. All right. So which of the following is not a signal that originates from a successful rearrangement of a beta chain and the pre-T cell receptor? So this is going into T cell maturation. Make sure you know so B cells and T cells, you've studied many chapters about B cells and T cells. So make sure you know their maturations, right? For B cell and T cell. The B cell, it's a little easier in my opinion. The T cell is a little bit more involved, but it's still similar to the B cell, right? Just to give you a quick recap, remember, you begin with a stem cell that goes from the bone marrow to the thymus. Then in the thymus, the stem cell, which is uncommitted, receives Notch 1. And because of Notch 1, it turns into a committed progenitor cell, right? So it goes from being able to turn into whatever blood cell into a cell that is committed now to differentiate into a T cell. So then it starts doing somatic recombination, the beta chain first, right, with its V, D, and J segments. then just like the B cell with its heavy chain, it needs to test that. So to test it, it's going to go through the like the pre-T alpha or pre-TCR check. And there, if it works, it's going to receive a signal that will, the question is asking which of the following is not a signal. So the signals are going to be stop B cell beta chain rearrangement, express CD4 and CD8 on the surface, start proliferation, and allow for alpha chain rearrangement. So basically, prepare the DNA so that alpha chain genes can be rearranged as well, right? So then after that, you go through the alpha chain, you make sure you have an alpha chain that works, and then, because you're a T cell and you need to recognize MHC, you need to go through positive selection. And in positive selection, you start testing your TCR against MHCs. And when you recognize an MHC, that's where you determine whether you're going to be a CD4 or CD8 cell. So basically, you're floating around, testing, and whenever you can bind to an MHC, if it's an MHC1, then you're going to turn into a CD8 cell. So all the CD4 that you had on the surface, because you know, right up until that moment you were a double positive cell, meaning that you had both CD4 and CD8. So if you recognize an MHC1, you keep all the CD8, you cut out and remove all the CD4. And then if you recognize an MHC2, then you keep all the CD4 and get rid of all the CD8, right? And then after positive selection, you go through negative selection, where you make sure that your maturing T cell or your thymocytes do not recognize self-antigens, right? So if you have a T cell that actually binds to self-antigens, you just get rid of that. And then the rest of the T cells that are able to go through the negative selection, then you let them free into circulation, and you have T cells that now work for you and will protect you against stuff. Nope, don't turn off. Make sense? Okay. So the correct answer here, so the signal that will not originate from successful rearrangement of the beta chain and the pre-TCR is expression of either CD4 or CD8, because you express both CD4 and CD8. Make sense? Yeah? Okay. So for 31, it's asking, once the first chain of the beta chain of the TCR is rearranged and the cell is finished proliferating, the RAG genes are turned back on, the arrangement of the alpha locus is induced. There's a bigger chance that the rearranging thymocyte will become gamma delta than alpha beta. A and B are all of the above. So the correct here is that this is similar to the first answer, the first question, not the first question, the questions before. But just remember that whenever you work on, even for B cells, if once you work on a beta chain, for example, you turn off RAG1 and 2, you test it, and then you turn back on RAG1 and 2, right? Same thing for the B cell. With heavy chain, you turn on RAG1 and 2, you work on the heavy chain, you turn it off, test it, and then you turn back on RAG1 and 2, and you work on the alpha chain. The same thing for the T cell. RAG1 and 2 on, beta chain, turn it off, test, turn it on again, alpha chain, turn it off, and you're done. Yeah? Cool. So 32, this is asking if the pre-T cell receptor is composed by a fully formed TCR with both alpha and beta? Yes or no? No, right? Because the pre-TCR is only the first chain made, so the beta chain. So this is wrong. The beta chain and the pre-T alpha, which is true. CD3 and the zeta chain, which is also true. So then the correct answer is E. So these. All right? All right. Rearrangement of the alpha chain locus continues throughout the process of positive selection and stops whenever a T cell is positively selected. This is true, and it's because, remember, the positive selection happens right after alpha chain rearrangement, right? And in positive selection, as a T cell, you're just testing whether you can recognize MHCs. So if you're a T cell and you're testing, you're trying to scan and trying to recognize something, but you're not able to recognize anything, you can still go back to your genes for the alpha chain and tweak them, try to make new rearrangements until you get to recognize something. Now, in some occasions, some T cells are not able to recognize anything, so then they eventually die by apoptosis, but they still have, a couple of chances to recombine and keep the somatic recombination going until they can recognize something. Okay? Yeah? Cool. All right, Tregs. Oh, I didn't fix this one. No, I did. Okay. So a Treg, it's a type of CD4 cell, right? It's a CD4 T cell. It's an effector CD4 T cell. So A is correct. And it expresses CD25, which is true, but this statement is saying that it's a transcriptional repressor. CD25 is just a protein that's on the surface of Tregs. So B is wrong. C is right because it says that it expresses FOXP3, which is a transcriptional repressor used by Tregs, and it's unique by these cells. So basically what Tregs do is they will shut down, they'll regulate other T cells and other cells in general. And the reason they're able to do that is because they can use this FOXP3, which activates special genes for these T cells to, do their job, basically. Just remember, FOXP3 is a transcriptional repressor, and they also have CD25 on their surface. But FOXP3 is the important one. All right. So positive selection tests whether, and again, CDRs. Which CDRs recognize the MHC again? 1 and 2, right? So positive selection tests whether the CDR1 and CDR2 region of the TCR, can bind to a MHC. So the correct one is 1, 2, MHC. Make sense? And then in negative selection, you're testing which CDR. Because you're testing whether you can recognize peptides, right? So you're testing whether the CDR3 can recognize self-antigens, right? If you do recognize self-antigens, that's when you tell that T cells to die by apoptosis. But you are testing the CDR3 region in negative selection. Positive selection, CDRs 1 and 2. So 36, this is asking that when a T cell is screening the peptide MHC complex on an APC, the T cells LFA1 can bind to ICAM1 and 2 on the APC. ICAM3 on the dendritic cell binds to DC sign on an activated T cell. And then SP1 receptors will be suppressed if the T cell is able to bind successfully to the peptide MHC complex. So, really quick, T cell activation. Remember, a T cell is going from lymph node to lymph node scanning antigens on dendritic cells, right? The dendritic cell is presenting the antigens on an MHC class 2. Whenever a T cell actually recognizes something on a dendritic cell, it's big, right? It's probably bad for you. You need to make an immune response against it. So what will happen is that the adhesion molecules on the T cell and dendritic cell will bind to each other so that the T cell and the dendritic cell stay together. So LFA1 and ICAM1 and 2, right? Then B is wrong, but because it's flipped. Because DC sign is on the dendritic cell, and it's DC-SIN is an adhesion molecule that's typical, and you can find it on dendritic cells. It's one of those it's one of those molecules that if you want to make sure it's a dendritic cell, you could test for the presence of DC-SIN. And DC-SIN binds to ICAM3. It's just that for option B, they're flipped. DC-SIN is on the dendritic cell, ICAM3 is on the T cell. So B is wrong. And then C is true because SP1, it's S1P. What did S1P receptor do? Or what did S1P do for T cells? Does anybody remember? So what did CCL19 and 21 do for T cells coming into the HEV? Does anybody remember? What's that? What did CCL19 and 21 do for incoming B cells or T cells? Yes, back there. Yeah, I told them to come in, right? Come on right in. We have antigens you need to scan, so get through this HEV. Now, let's say T cells get into an HEV and they're in the lymph node, but a T cell already recognized something, so there's no more dendritic cells available to show stuff. And all the rest of the T cells, they need to leave and they need to go somewhere else because there's nothing else in there to be, scanned. How does the dendritic cell tell the rest of the T cells go away? S1P, right? I misspelled it here. This is supposed to be S1P. But S1P is like sphingosine 1-phosphate or something like that. They're just the opposite of CCL19-21. They're telling the B cells and the T cells go away instead of come in. And an activated T cell that recognized the peptide presented by the dendritic cell, it's going to get rid of its S1P receptors because you don't want that T cell to receive the signal of leave this lymphocyte because you want that T cell to stay, start dividing, get activated, and all that stuff, right? So the S1P receptor will be suppressed, they'll be removed so that the T cell can bind successfully and then get activated, basically. Okay? Cool. Where is it? So, which of the following statements regarding the co-stimulatory protein B7 is true? So remember, B7 is the one protein that's expressed by dendritic cells, right? And basically, it's the one protein that if it's not there, the T cell will not get activated, right? Because B7 is expressed only when a dendritic cell takes in something that's pathogenic, that is exogenous, that's not from your body, right? So if a T cell recognizes whatever the peptide on the MHC and B7 is present, then good. You have activation, you have an immune response. Now, dendritic cells can pick up things that are, from our own body and still take it to T cells. But if, let's say, a dendritic cell picks up a dead epithelial cell and then it goes to the lymph node and it shows it to a T cell, there's not going to be any B7. So if a T cell recognizes our own epithelial cell and B7 is not present, then the T cell is told to deactivate, to go into the anergic state, and then die eventually. Yes? So then the correct one here is going to be C. Just to make it a little faster. A is not true because CTLA4 has a 20-fold binding strength more than CD28, and this one says less. And then B is wrong because B7 is not on the surface of the T cell, it is on the surface of the dendritic cell, and then CD28 is on the surface of the T cell, not of the APC. Then 38, oh Lord. A T cell that recognizes self-antigen will most likely be rendered anergic because of the, no, B is wrong. Okay, disregard this question because this is, the wording will make you more confused. Don't even look at 38. This is a question I was supposed to remove and I didn't. Because the way I wrote it will make your brain blow up at this point. Because this is question 38. Yeah, I'm going to remove it. I'll re-upload everything and I'll make sure to remove it. But people who are watching home, I guess, from YouTube, if there's anyone, just don't read question 38, okay? Cool. Also read it here. Do not read. Okay. Sorry about that. So all leukocytes are lymphocytes? False, right? All lymphocytes are leukocytes? Yes. Cool. IL-4 will induce differentiation of which type of T cell? TH? What's that? 2. Yes. Awesome. So I don't know if you need to know, like, so for test, I want to say 4, you needed to study the chart where you had, like, TH1, TH2, TH17, and if you have these cytokines, then you'll get a TH1, which will secrete these other cytokines. I don't know if you need to know those cytokines, but at least try to study the more important ones, right? So IL-4, the cytokines that tell lymphocytes to come in and out. So CCL19-21, CXCL13, CCL25. IL-4 definitely, because he's talked about IL-4 a few times. So I guess this one is good to know. But if you can remember which cytokines induce which T cells to proliferate, that's always a plus for you. Yes? 8, I want to say. I think it's 8, yeah. Cool. So lepromatous leprosy, that's another of the diseases that you need to study, right? So lepromatous leprosy is the high infectivity one, because you had two, tuberculous and lepromatous, right? The difference between the two is that they will cause a bias of differentiation. So lepromatous will cause a bias towards Th2 cells, right? And then tuberculoid will cause a bias towards Th1 cells. Now remember, leprosy is a bacteria that infects the vesicles of your macrophages. So basically it inactivates the macrophages that are there because they're supposed to help you out in phagocyte stuff. So the lepromatous is the high infectivity one because it causes a bias towards Th2. And when you have Th2 activation, you'll get, basically you have activation of B cells. And B cells are not going to be any good if you have a bacteria that's inside macrophages. Because if you make antibodies, these antibodies are not going to be able to get into the macrophages, right? So the lepromatous leprosy that has a Th2 bias is worse because of that. You just create an immune response that's not going to work. With tuberculoid leprosy, you have a better chance at fighting it because you have a Th1 bias that activates more macrophages. So eventually you get activation of macrophages that have not been infected, and you can slowly fight off the infection. All right? I know you've heard me speaking for an hour and 38 minutes. So remember, this is all recorded on YouTube, so if you didn't understand me, you can like take a break later and listen to like the second half. But does it make sense? Yeah? So the correct answer here is B and C. So it's a high infectivity disease that causes differentiation and bias for Th2 cells. And then the cytokines present there are going to be IL-4, 5, and 10. For macrophage activation, all right. For macrophage activation, there's two signals that you need. When, for example, when the Th1 cell activates the macrophage, there's two signals needed. The first one is CD40 ligand interaction. CD40 is on the macrophage, CD40 ligand is on the T cell. And then the second one is that Th1 T cell secreting interferon gamma that then binds to the macrophage. All right? So then what you need is, yes, this one. Boop. All right? Make sense? Yes? Okay. Yes? CD40 on the macrophage, the ligand is on the T cell. So remember, where else did you see CD40 ligand? Do y'all remember? Which topic? B cell activation? Remember, when the B cell came out of the follicle after somatic hypermutation and isotype switch, you needed CD40 ligand interaction. CD40 is on the B cell, CD40 ligand on the T cell, so that the B cell gets the survival signal, BCL-XL, and it can live, basically, right? So CD40 is present in many cells. CD40 ligand is on the T cell all the time. And then with hyper-IgM syndrome, which is another disease that you went over during the semester, you have a lack of CD40 ligand on the T cells, right? Because of a mutation or whatever problem you have that doesn't let you express CD40 ligand. So then the B cells are not able to go through somatic hypermutation and isotype switch, and the only thing you can make is IgM. Therefore, hyper-IgM syndrome. Yeah? Cool. So when an activated naive B cell starts interacting with a T follicular helper cell, so just to make it quick, the correct option here is all of the above. So the point of this question is to help you review the important things that happen when a naive B cell is basically activated by a T cell. So you'll have release of IL-5 and 6 by the T follicular helper. IgM will be the first thing produced, and it's going to be the most abundant antibody for that infection. And then the B cell will form this conjugate pair with the T follicular helper. You're going to have CD40 expression of CD40 ligand, activation of NF-kappa B. If this doesn't ring a bell, just go back and review the B cell activation chapter. Okay? Oh, Lord. Cool. So which of the following statements is true regarding a recently somatically hypermutated and isotype switched B cell? They'll have to compete to interact with the T follicular helper. Will die by apoptosis if the B cell is induced to produce the BCL-XL signal, and it can turn into a plasma cell or a memory cell without signals from the T follicular helper. So just to give you a quick review, when a B cell is in the follicle going through somatic hypermutation, it's going to turn into a central blast first, where it's actually doing the mutations, and then it's going to go in the light zone where the centrocyte is, and it's going to compete with other B cells to pick up some antigens from a follicular dendritic cell, right? Whenever the somatically hypermutated centrocyte, or B cell, picks up the antigen, it's going to take it in and then put it on MHC class 2, and then it needs to go out of the follicle to the T cell and present it to the T cell. And when it gets there, the T cell, will recognize the MHC2, and the T cell is also going to have CD40 ligand that binds to CD40 on the B cell, and that's the survival signal I just said earlier, so that the B cell can live. The survival signal is given by BCL-XL. So the correct one is going to be A and C. Yes? Make sense? Any questions so far? All right. So this is more, the 45 is another question about the signals that T cells give to B cells during an infection. So when an infection just began and we have an increasing amount of pathogens, the T follicular helper are expected to secrete more blank to induce B cells to differentiate into blank. So if you have an infection, you need more B cells activated into plasma cells, right? Which cytokine will you secrete to tell B cells to turn into plasma cells? Is it IL-4 or 10? Does anybody remember? Anyone? 4? Yeah, right? You ever remember that? IL-4, make plasma cells, or turn into a plasma cell, and then IL-10, just turn into a memory cell, we'll use you later. All right? Plasma cell. Cool. This question goes back, 46 goes back to complement. So make sure you study complement. When you study complement, make sure you also study the regulation of complement. So make sure that you know what factor P does, what H and I do, and then what MCP and DAF do. It's like in this complement slide, there's like a little table that shows you all of that, okay? Because part of knowing complement is the actual complement cascade, but you also need to make sure that you know the regulation. So this question is about the regulation, and it's asking which of the following regulatory proteins are used to slow down the activation of complement. So factor P, remember, is the one that actually accelerates complement. It stabilizes the C3 convertase, so that's not A. DAF and MCP, those are two that will slow down complement. And then the other two is factor H and I that are not here, but just so you know. DAF and MCP, those are slowing down complement when it's activated in your own cells. And then factor H and I will slow down complement in, bacteria, virus, wherever else that's not in your own cells, basically. Is there any other questions so far? No? Okay. Cool. So which of the following scenarios can be considered as a form of passive immunization? IgA crossing the placenta to enter the fetus? Is that true? No, IgG crosses the placenta, not IgA. Okay? So somatically hypermutated and isotype switched IgG that's being released by a plasma cell in a secondary lymphoid tissue during an infection. Is that passive immunity? No, right? Because remember, passive immunity is getting antibodies from somewhere else other than your own B cells. And B is describing your own B cells secreting antibodies, right? C, Steve Irwin getting antivenom after being bit by a black cobra because he felt like petting the thing, right? That's a form of passive immunity. Another form of passive immunity, IgG crossing the placenta to the baby, or IgA being given to the baby through breast milk. Yes? Cool. So the correct one here is C, so that I can just read it out loud. So naive lymphocytes can circulate through the malt, lymph node, spleen, or any secondary lymphoid tissue because they follow a CCL 19 and 21 gradient when entering the tissue through the HEV. But then when a lymphocyte is activated in the malt lymphoid tissues, they will only follow a gradient of CCL 25 and then they will express an adhesion molecule that's alpha-4 beta-7. You should know this. You just studied this for the last test, right? This is easy. Then when an individual has a helminth infection, the more effective response is going to be Th1, secreting interferon, secreting interferon gamma. Because remember, Th1 responses will allow you to secrete mucus, activate eosinophils that are really good for parasites. They'll be able to activate macrophages without activating inflammation. So it's all things that will not damage your GI tract, whereas a Th2 response actually it activates inflammatory response, and it's just more damaging to your GI tract. And then what's the receptor for IgG that transports IgG? There's 3 names for it. Bramble, FcRn, and FcRb, right? So all of the above. Boom. What's that? Oh, sorry. Nope. All of the above. All right. Any questions? So for those of you who came in, so I'm guessing like it's mostly people, so if you can just pass these down. Make sure you do this review survey. You need to put in your UCF ID and bubble in session 2-6. Thank you for coming today. I really appreciate it. Good luck on the final. If you still are not sure like which topic to focus on, try to focus on those big topics that we went over today that you can find on this review. B cells, T cells, somatic recombination, and the innate immune response. Don't get bogged down on like the tiny details. I did have a few tiny details here and there just because when I took the final, I remember there were a few, and it's just good to know some things specifically. But divide and conquer. Make sure that you understand the big ideas first, the big concepts that you went through the whole semester, and then try to focus on the little things. Okay? If you don't know how to review, like if you still are not sure, maybe try to go over the practice exams I had. You can try to go over the study guides also. Try to answer each study guide because it has a bunch of questions in them. If you don't know how to answer one question, then just go back to the PowerPoint, look up the information, and then answer it. And then make sure you get a good night of sleep before. So if you want to do like a cramming session, just do it 2 nights before, and then the night before, just go to sleep early. Because, you'll be able to recall information much better if you actually had a good night of sleep and you're well rested and you feel good, right? If you only slept like 2 hours and you feel like crap and the caffeine hasn't kicked in yet, then you're setting up yourself for not succeeding, right? Does it make sense? Cool. Well, thanks for coming. Thank you for all of you that came to my sessions during the semester. I appreciate it. And I'll see you during the final exam. All right?